All right. Butterflies, those dazzling creatures with wings that look like living stained glass. They're just so captivating.
Yeah.
And I think we all kind of know that genes are involved in making those intricate wing patterns.
Mm-hmm.
But get this, it turns out there's this tiny molecule. It's called microRNA.
Oh, wow.
And it's totally changing how we think about how those colors come to be.
Really? So it's like, what, a secret code or something?
Kind of like that, yeah. It's like a secret code hidden right there in the DNA.
Wow.
So I bet you're wondering, what exactly is microRNA, right?
Yeah, I am actually.
Well, just imagine it as a dimmer switch for genes.
Okay.
It's basically fine-tuning how much of a gene's instructions actually get carried out.
I see.
And, for a long time, scientists thought microRNAs were just these tiny players in the whole scheme of genetics. But now they're starting to think they might be like the master conductors of the orchestra.
Interesting.
Speaking of intricate designs, that British peppered moth?
Yeah.
Total classic example of natural selection.
Right.
During the Industrial Revolution, those darker moths, they had a survival advantage.
Right, because they blended in.
Exactly. They blended in with all those soot-covered trees.
Yeah.
While those lighter moths, they really stood out and became easy prey.
Makes sense.
So it makes sense why it happened.
Yeah.
But the how has always been a bit of a puzzle.
Yeah, that's true.
Until now.
Oh.
Today's deep dive is taking us into this really cutting-edge research, and it's revealing just how powerful microRNAs can be. This paper, it was published in Science, focuses on this specific microRNA. It's called miR-193.
miR-193.
Researchers found that this tiny molecule is actually playing a crucial role in determining whether a butterfly's wings are dark or light.
Oh, wow.
For a long time, scientists thought this gene called cortex was the main driver of black and dark wing colors in butterflies and moths.
Yeah, that seems to make sense.
Yeah, it seems logical, right. But here's where it gets interesting.
Okay.
This researcher named Shen Tian, he was studying at the National University of Singapore.
Okay.
And he decided to test this theory.
So how do you do that?
He used this technique. It's called gene knockout.
Oh, I've heard of that.
Yeah.
Yeah.
It's essentially turning off the cortex gene just to see what would happen.
Oh, clever.
He expected those dark wings to turn light,
Yeah.
But to his surprise, nothing changed.
Huh.
So that's when it gets interesting. What made him suspect that microRNAs might be involved, even though that cortex knockout didn't work?
Well, sometimes it's just scientific intuition, right?
Yeah.
I think he had this hunch that something else was regulating the cortex gene, and microRNAs were, kind of a prime suspect.
Yeah.
The challenge with them, though, is they're really difficult to study.
Oh, how so?
Well, they're tiny, and they don't show up easily in the ways that scientists analyze gene expression.
I see. So how did he manage to find these hidden molecules, it sounds like, trying to find a needle in a haystack?
Right, but a microscopic needle.
Exactly, yeah.
Tian, he was determined, though.
Good for him.
He actually created his own special microRNA libraries.
Oh, wow.
Specifically for butterflies. Talk about dedication, right?
Yeah.
And through this really meticulous work, he discovered miR-193 and another microRNA called miR-2788, and both of those were located near the cortex gene.
Interesting.
Yeah, this was a big clue.
Yeah.
But there was still a lot of skepticism in the scientific community.
Why is that? Weren't they already starting to realize how important microRNAs are?
Yeah, they were.
Yeah.
But they were often seen as just these fine-tuners, not major players,
Uh-huh.
So Tian's hypothesis that a microRNA could be the primary regulator of something as big as wing color, that was pretty bold for the time.
Oh, I see. So how did he prove his point?
Well, he used this revolutionary tool. It's called CRISPR-Cas9 gene editing.
CRISPR.
Yeah. Think of it like a molecular scalpel.
Okay.
It allows scientists to make these incredibly precise edits to the genetic code.
Oh, wow.
And in this case, Tian used it to knock out miR-193 and miR-2788 in 3 different butterfly species.
Wow.
Pretty risky move, but the results were stunning.
Okay, I have to admit I'm on the edge of my seat. What happened when he knocked out miR-193?
The butterfly's wings changed color.
Oh, wow.
They became lighter, just like he predicted.
Wow.
It was a defining moment, irrefutable proof that this tiny microRNA was actually a key player in pigmentation.
That's incredible. How does a microRNA, something so small, actually control something as complex as wing color.
Yeah, that's where it gets really fascinating. miR-193 isn't just flowing around on its own.
Right.
It actually originates from this longer non-coding RNA called ivory. And miR-193's target, a gene called ebony. And that gene is known to be involved in melanin production.
Wait a minute. Melanin. Isn't that the same pigment that gives color to human skin and hair?
Exactly.
Yeah.
It plays a role in pigmentation across a wide range of organisms.
Wow.
And what's remarkable is that Tian's research is suggesting that a tiny microRNA is essentially pulling the strings.
Oh, wow.
It's regulating how much melanin is produced in butterfly wings.
That's mind-blowing. And if I remember correctly, the ivory gene itself was already linked to butterfly wing patterns.
Yes.
So this discovery is adding a whole new layer to our understanding. It's like we've zoomed in on the genetic code and discovered a whole new level of detail.
Absolutely. And, you know, that's just the beginning. This research opens up a whole Pandora's box of questions about the role of microRNAs in other organisms.
I was just thinking, could miR-193 or a similar microRNA be influencing colors and patterns in other insects too? The possibilities seem endless.
They really do. Think about the incredible diversity of insect life, all those intricate designs.
Yeah.
And now we have a whole new avenue to explore.
Yeah.
We might even find that microRNAs are influencing traits beyond just pigmentation.
That's incredible.
Yeah.
It's amazing, right? These tiny microRNAs, they might have a bigger impact than we ever realized.
Yeah, for sure.
What I think is really fascinating is how this discovery connects back to that, classic example of evolution, the peppered moth.
Yeah, it is.
Remember how we were talking about those darker moths thriving during the Industrial Revolution?
Right, yeah.
Well, now we might actually have a possible explanation for how those color changes actually happened, like at the genetic level.
That's interesting.
We understood, right, the environmental pressures that favored the darker moths.
Yeah.
But the actual mechanism behind the color change was kind of a mystery.
Sure.
So could microRNAs have actually been, like, pulling the levers behind the scenes?
It's possible.
That's what I'm thinking.
Uh-huh.
What if, right, as the pollution was darkening the trees, there was all this strong selective pressure for moth that had those darker wings?
Right.
So maybe subtle changes in microRNA expression, maybe even a microRNA like miR-193, allowed some moths to produce more melanin.
And that gave them that camouflage advantage.
Exactly.
Yeah, it makes sense.
So instead of waiting for a random DNA mutation to kind of slowly spread through the whole population, microRNA could have provided this faster track for adaptation.
Yeah, like a shortcut.
Yeah.
Totally.
It's like having a volume knob for gene expression. So if the environment changes, microRNAs could allow organisms to quickly adjust their traits without even altering that underlying DNA blueprint.
That's amazing.
It's a fascinating example of how evolution might work on a much faster timescale than we previously thought.
Way faster.
Yeah. Now it really makes you think about the implications for other species.
Yeah, definitely.
Including humans.
Yeah.
Could changes in microRNA expression have actually played a role in the diversity of, like, human skin and hair color?
Oh, that's a good question.
Right?
Yeah. I mean, we already know that melanin plays a key role in human pigmentation.
Yeah.
And if microRNAs are regulating melanin production in butterflies, it's totally reasonable to assume they might be doing something similar in us.
It's incredible to even think about. But let's get back to those butterflies for a minute, though. You mentioned that Shen Tian, he used CRISPR to knock out miR-193.
Uh-huh.
Can you imagine that feeling when he saw those light brown butterflies emerge from their pupa?
Oh, it must have been incredible.
I know.
To see, like he had this hunch, this bold idea that kind of went against, the prevailing wisdom.
Right.
And then to actually see it confirmed.
To see it right there in front of him.
Oh, I bet that was so cool.
I know. It's a testament to, the power of scientific intuition and perseverance.
Totally. And the implications of his work, they go way beyond just butterfly wings, too.
Yeah, for sure.
His research has opened up this whole new perspective on how traits are controlled.
Yeah.
Not just in insects, but potentially across the entire animal kingdom.
It's like realizing that we've been looking at the genetic code with one eye closed.
Totally.
And now, with the discovery of microRNA's influence, we're finally seeing that full picture.
Yeah.
That intricate interplay between genes and their regulators.
And think about the potential applications.
Yeah, it's huge.
If we can understand how microRNAs control pigmentation, that could lead to new ways to treat skin disorders.
That would be amazing.
Right. We could develop new types of dyes and pigments for textiles or even art.
Wow. I never even thought about that. It's amazing to think that something so tiny could have such a huge impact, both in the natural world and in our own lives.
Right.
But before we get ahead of ourselves, let's get back to the basics for a moment. For our listeners who might not be totally familiar with microRNAs, could you explain a little bit more about how they actually work?
Yeah, sure. It's important to remember that microRNAs are not directly altering the DNA sequence itself.
Okay.
Instead, they're acting like this complex network of switches. They're controlling which genes are active and to what degree.
Just like a dimmer switch.
Yeah, exactly. It's a little bit like having a dimmer switch on a light. Instead of just turning it on or off, you can fine-tune the brightness.
So unlike a mutation, which permanently changes the DNA, microRNAs offer a way to kind of fine-tune gene expression on the fly.
Precisely.
Uh-huh.
And that makes them incredibly powerful regulators of development, adaptation, and even evolution.
That's amazing. You know, it's incredible to think that something so tiny, something we really didn't even know about until recently, could have such a huge impact on the way life actually unfolds. It makes you really appreciate how complex and elegant nature's design really is.
Yeah, it's humbling for sure.
Absolutely. And speaking of these tiny details, let's kind of zoom back in on the mechanism behind miR-193. I'm still trying to wrap my head around how it actually works. You mentioned that it targets a gene called ebony, and that's involved in melanin production. Can you kind of walk us through that process?
Oh, yeah. So you have the miR-193 molecule, and it's actually produced from this longer RNA strand called ivory. This tiny molecule is acting like a homing beacon. It's seeking out the messenger RNA that carries the instructions from the ebony gene.
Okay, and then what happens when the miR-193 finds its target?
Well, it basically blocks those instructions from being carried out. Remember that analogy we were using with the dimmer switch?
Right.
Well, in this case, miR-193 is turning down the volume on the ebony gene.
Oh, okay.
It's reducing its ability to produce melanin, and less melanin means lighter wing colors.
Oh, I see. So it's like the peppered moth's color shift might have been driven by microRNAs, kind of tweaking that same melanin pathway that we're seeing in these butterflies. It's like these totally unrelated phenomena are suddenly connected by this like tiny molecular thread.
Yeah, and that's what makes this research so exciting. You know, it's not just about butterfly wings.
Right.
It's about understanding those fundamental mechanisms that could be working across so many species, driving evolution and shaping that diversity of life that we see all around us.
It's like we've kind of stumbled upon this hidden language of genetics, a whole new level of control that's been going on right there beneath the surface all along.
And the research on miR-193, you know, that's really just the tip of the iceberg. Scientists are now exploring all the roles of these other microRNAs in a bunch of biological processes, you know, everything from development to even disease. It's a whole new frontier of scientific exploration.
Makes you wonder what other secrets are hidden in the genome just waiting to be discovered. What are some of the big questions that scientists are trying to answer about microRNAs right now?
Well, one of the big ones is we're still trying to map out all the different microRNAs that even exist and then figure out what their target genes are.
Right.
It's a massive undertaking, you know, like trying to decipher a complex clerk.
And then beyond that, how can we use this knowledge to actually understand and maybe even manipulate those biological processes?
Well, that's the million-dollar question, isn't it?
Yeah.
Imagine all the possibilities if we could harness the power of microRNAs to treat diseases or engineer crops that have certain desirable traits.
Yeah, it's a field with so much potential.
It is, but it's still super early.
It's exciting, and it's a little daunting to think about all those implications. But I think before we get too far into the future, let's appreciate how far we've come. Just a few decades ago, we barely even knew that microRNAs existed, and now we're starting to really unravel their secrets and kind of glimpse at their incredible power.
Yeah, for sure. It's a real testament to the power of scientific curiosity, you know, and how important it is to invest in this fundamental research. Who knows what kind of amazing discoveries await us as we continue to kind of explore this hidden world of microRNAs.
Well, that is so true. And on that note, I think it's time for us to wrap up this deep dive.
Yeah.
It's been a pretty incredible journey, don't you think? Exploring the world of butterfly wings, microRNAs, and all those surprising connections that tie them all together.
It has been a pleasure.
And to all of our listeners, we really hope this deep dive has sparked your curiosity and left you with a sense of wonder about all the hidden complexities of the natural world. Keep exploring, keep questioning, keep learning, because there's always something new to discover.